Hello again, and welcome back. We get to part 3 of modeling the determinants of growth for an insect population. So in this talk, we've looked at so far 2 stages of a research project. We looked at the way that the original hypothesis and rationale for the experiment was set up and how the experiment was conducted, and we looked at the analysis of the results. And we saw that we could use simple descriptive techniques, graphs, tables, statistical analyses to confirm the hypothesis that odor-baited insecticide-treated targets could be used to control tsetse. We then asked whether it was possible to model the changes in fly population as a function of climatic variables, trapping pressure, and found that yes, we could get a good fit of the data, suggesting that temperature was the most important climatic factor influencing tsetse population dynamics. And that's an important result. People hadn't been able to demonstrate that before. So what we've done, let's think about what we did. We followed cohorts of flies using a deterministic process, and we know that actually it would have been better, or possibly better, to follow individual flies and to make the simulation process entirely stochastic. But the simulation would have taken longer. Small changes in parameter values led to relatively large changes in the residual sums of squares when we actually did try to do that. And we saw large numbers of local minima in the loss function that made it impossible to calculate derivatives. We could have overcome this by using a simpler minimization technique, but this would have been very slow. But on the other hand, this could be compensated with faster, more modern computer networks and networks. The point is that there's more than one way of getting the same results. So what I've shown you is one particular way, but it's not the only way that can be done. Now, having done the modeling and got the results and seen what actually happens, what are we going to do? What are the graphical outputs that will display the answers to the question? So, I mean, what I've just shown you is what we've actually done in terms of fitting the model. But what we want to do is we want to just have some nice things which will actually show the reader what's actually going on. So graphs, for instance, of the observed and predicted total population, also the observed and predicted births and survival probabilities, which I didn't show you. But all of those we've got very good fits to, so you want to do that. Now then, also, when you're actually, the thing to remember, in fact, when you're doing the modeling itself, is that it's an extremely good idea to write comments in your code as you're going along and to be debugging it as you go along. This has been a very rapid gallop through this procedure, but the fact of the matter is that the Fortran code that we used for doing this thing is a couple of thousand lines long, and you can get very, very severely lost if you don't actually make comments to show you where you're going. And if you do that, it'll make it much easier for you to find errors, because trust me, there are always going to be lots of errors as you go along. And of course, what you're going to be doing is running this model tens, hundreds, perhaps thousands of times. So you're going to have huge numbers of versions, and you need to use something like Git or some other thing to keep a track on where your versions are, so you can go back and see, okay, well, back there somewhere I ran something, and what does it look like? And then finally, what you should be doing is doing simulations, actually, which will verify your methods and help you to debug, because we've been doing simulation anyway, but we need to make sure that those outputs make sense, and doing a simulation of the thing actually really will help you to see that. And it's an extremely good idea, as you're actually writing things up, to write down the methods at the time when you're actually doing the modeling. Write down the methods so that you can figure out what is going on. Now, one of the things that we haven't talked about is a sensitivity analysis. The particular method that I used is just a program called Search, which was written in Fortran quite a long time ago. And it's a very sophisticated fitting routine. And it provides errors for each of the parameters. So the parameter, for instance, for the effective targets or traps or the effective temperature, when you've got the coefficient, it'll give you an error for that coefficient. And which that'll tell you, if you go through those, say which is going to be the most important, which is going to be the most important variable, parameters involved in fitting things. And you can actually do some sensitivity analysis by varying, for instance, the initial conditions. So we know that Vail put 4,000 pupae on the island to start with. What if we change that to 2,000 or 8,000? Would it make any difference to the way things go on? It shouldn't do. What it should be is that ultimately things will settle down. But if it is a big, strong function of the initial conditions, you might have a problem. And also, you could do things like vary the climatic inputs. All we know for the temperature is what we read in a Stevenson screen, some met data screen, and that may not be the temperature which the flies are actually experiencing. So what happens if we actually change the temperature that they're supposed to be living at? Does that make a difference to our model fit and that kind of thing? Then also it's a good idea to do some model validation. So for instance, let's suppose what we did was we followed what happened to we know what the population was actually doing when targets, embedded targets and traps were put on the island. But suppose those targets and traps had not been put on the island, then it better be true that the population would not have died off in the way that we actually saw it do. And we can check that by just simply running the simulation, but actually not putting in any traps and targets, and see then how it changed the way in which the population grew. You can also look, and it's be a very good idea and if other people want to actually use, for instance, an ordinary differential equation approach to fitting the data, do they get results as good as the ones we've actually found? And what I see so far is that nobody's managed to come up with an ODE approach which actually produces anything like as good a fit to the data. So that's something else to look at. Right. When it comes to writing up the results, make sure that you state your assumptions clearly. And the most important thing is critique your own work, and critique it as if you were a reviewer, a nasty reviewer who's actually looking for holes to pick in it. So pick holes in your own stuff, because that'll save you a lot of time and stress later down the line. Then it comes to the matter of deciding where we're going to publish this work. And this is a difficult one. Yeah, difficult one. And you need to get real. Of course, you think your paper is absolutely brilliant. It should be in Nature, and that's really great. But it may very well turn out that actually nobody really cares too much about tsetse flies, and that I can tell you from experience that is actually the truth. And you may yourself be working in an area which is not flavor of the month. And so you can actually waste an awful lot of time and get a lot of pain battling with high impact journals when they're simply not going to take your paper. The most important thing, or an important thing in this thing, is to get this paper off your desk and get on with your life, get on to the next study, get on to your next paper. For the good of your career, of course you want to publish in high impact journals, but you also just need to publish, full stop. Period. So it's a balancing act. Aim as high as is realistic, and aim low enough to get it off your desk. That's my advice at any rate. When you're thinking more practically now, regardless of impact of one thing or another, when you're thinking about it, what are you citing? So with this particular paper that we talked about on Antelope Island, we're talking about stuff on tsetse. Okay, so that decides some of the journal that you might actually want to work on it, to put it in, where other people have done stuff on tsetse, for instance. On the other hand, if you've got a paper where, yes, it is on tsetse, but in fact you put it in a much more general context, then you can aim higher, and you can go for a higher impact journal and a journal which is concerned with more general matters of biology. And the way that you can actually look at this is to look at the statements, the journal, the scope statements on their website, and then look at other articles in the journal to see, look, does my article fit in here? Is it as good as these ones? Is it as interesting? And is it on the same sort of subject area as these ones? Then think about, who am I aiming at? Who do I want to actually talk to here? It's all very well writing this paper, but who is actually going to want to read it? And then you need to think in terms of, how mathematical is my article? Because a lot of journals are really not interested in articles which have got an awful lot of mathematics in it. On the other hand, some of them really are. And if you're, then you need to go for that. And then make sure that you look at how many, how long can the paper be? What's the maximum limit for the number of figures? What's the maximum number of tables? All of these things you need to take into account. Just a final thought on the actual modeling. As I say, the search was written by somebody at UCLA, indeed in the late 1970s and the 80s, in Fortran, which is now somewhat outdated, or at any rate unfashionable. So you might want to think in terms of using more modern languages like R or C++ to do your modeling. Whatever you use, though, if you're doing this kind of fitting, you want to use something which is using it used to be able to calculate first and second derivatives of what's actually going on to allow you to get rapid fitting, so that you can get major power. Compromising on this matter would reduce the power of the minimization technique. So just think about that. And right at the end, and going back to the questions that were raised by the students in part one, so having done all of this, now let's actually think about the answers to that person's question. So the question, the first one was, what kinds of data are useful for modeling? And the answer is, at least an answer is, carefully cleaned data from well-designed, well-executed trials and experiments. How to collect and access the data important in modeling, etc. So in this approach, we've actually stood this question on its head. We did not start with the model and then look for data. We started with the data set. We looked at it. We cleaned it. We played with it. We thought about it. We drew graphs. We drew tables. We fitted mathematical functions to it. We carried out statistical analysis. We tried various ways of interpreting it. And then, and only then, we derived a model. And only because we needed a model. And this is what I really want to stress above everything else that I've told you. Start with the data. Don't start with the model. Think about a real problem in biology and the way that you're going to crack that problem. Don't actually think that you're going to create that answer by looking at a model first. And finally, when is it okay to take data or parameter estimates from other studies and use them in my model? Use anything that you can lay your hands on. As long as it makes sense, and as long as you attributed the use in a transparent manner, then everything is up for grabs. So the last question was, is that if DAIDD is supposed to be for epidemiology-oriented people, be helpful to know how we can actually use our epi methods to contribute to model development. So my presentation, I hope, tried to show our use of standard classical epi techniques critical to getting to a basic understanding of what is going on. This suggested the kind of model required to improve that understanding. But first important step is the application of basic epidemiological techniques. And just to wrap it up, and the summary is what I'm basically saying: work with the best possible data that you can find. Look at and play with those data. First, use simple graphical approaches, then statistical methods. Go to dynamic models only when these will tell you things you cannot find using simpler approaches. Let the data drive the model, not the other way around. Be lazy. Look for simple solutions. Only add complexity when essential. And think. At every stage, think. And above all else, enjoy. Otherwise, there's really no point. Thank you.